For our project, we decided on polymerase chain reaction and how it relates to the basic concepts of molecular biology. Breaking down PCR, it is a process that is similar to DNA replication that uses a specialized medical machine known as a thermal cycler. Since it's similar to replication, researchers can use small samples like hair, saliva, or dried blood to create copies of DNA for analysis. They can use this technique for various uses, like the human genome, which is all of a person's chromosomes that contain genes that give us certain traits. This technique can also give us gene locations to track down a certain trait or abnormality. This is also used during crime scenes for forensics to create more evidence than what was found. And lastly, the technique is used for pre-implantation embryos and infection control, which we will discuss later. In this image, the first stage is denaturization, where when heat is applied, the DNA unwinds into two new strands. Then, as the temperature cools off in the machine, the two primers of DNA, which are small segments that complement the separated strand, attach onto this DNA. This step is called annealing. Then, in the last step, an enzyme called TAQ polymerase acts as like a glue and cements the primers and nucleotides, which are what makes up DNA sequence, makes it unique, all together to form a new strand. This process can then be reported in many times to create as many copies as needed.
I found an article on how polymerase chain reaction helps with the pre-implantation of embryos. Using PCR allows screening for genetic abnormalities, mitochondrial mutations, and chromosomal mutations. PCR happens after a biopsy to obtain the oocyte. Multiplex PCR is one method used for determining these abnormalities. Multiplex PCR has been recently used for chromosome copy analysis. Chromosome copy analysis helps detect variations that may result in disease or illness. The picture on the right shows a multiplex reaction. This image shows that in a multiplex reaction, all four primers go into one tube, and from there they are separated into the different genes. The results from using PCR before the implantation of embryos are that it has shown improvements in live birth rates and IVF cycles. Also, it has been determined that using PCR is reliable for determining abnormalities in a chromosome or gene, but it is not the best for trying to find differences in structural chromosomes. The picture on the left of the slide just shows an example of how PCR is able to detect abnormalities in this study.
A really interesting application of PCR genetic technology was noted in a study from the American Journal of Infection Control. The authors wanted to address the difficult management of gastroenteritis present with long-term and rehabilitation care facilities. Gastroenteritis caused by Clostridium difficile and norovirus are a major cause in the development of hospital-acquired infections. Their goal was to evaluate the contribution of a new two-step laboratory-triggered strategy based on successive PCR assays. And PCR assays are just the actual tests that they would use. Specifically, they wanted to know how efficient and helpful the PCR assays would be in early detection of the infectious agents Clostridium difficile and norovirus. And these are just pictures of the actual PCR assay technology that the authors would have utilized in the study. After the researchers compared the new strategy's outcome with the former standardized outcomes, the new PCR strategy identified 14 more patients with infectious agents. This caused the healthcare team to move the infected patients to contact precautions to prevent spread of infection. The importance of this journal article is the extremely beneficial use of PCR in the healthcare field. By identifying microorganisms by their own unique genes at the cellular level, we can use PCR to prevent against infectious diseases and improve the healthcare setting in a practical manner.
PCR has a very important role in the nursing practice because it can be used for diagnostic purposes, detection of genetic mutations and infection, DNA profiling, and genetic fingerprinting. PCR is of great significance to the nursing practice because it can assist in step 2 of the nursing process, the nursing diagnosis. PCR can help us as nurses identify the medical illness, potential problems, and make clinical judgments, which help advance our goal in problem solving. PCR has a substantial effect on interdisciplinary care. In an interdisciplinary team, a nurse has direct care with the patient, so we can provide important information when it comes to counseling for genetic health. Nurses are able to bring a lot to the table with PCR when it comes to collaboration with other healthcare professionals in planning care, leading to positive outcomes in genetic health.